Enterobacterales. So patient is a 71-year-old male hospitalized for diabetic ketoacidosis. Patient complains of flank pain and painful urination, urine for culture. It's grown on McConkey, moderate lactose fermenting organisms. Non-hemolytic oxidase positive. It's indole positive, methyl red positive, Voges-Proskauer negative, and citrate positive. Other biochemical reactions, H2S negative, urea is hydrolyzed, arginine and ornithine are both decarboxylated. Antimicrobial susceptibility testing is displayed here. So setting the stage, comprehensive genomic analysis with phylogenetic reconstructions of the members of the heterogeneous family Enterobacteraceae resulted in the creation of a new order, Enterobacterales. So we used to always call them Enterobacteraceae. Now we're calling them Enterobacterales. All members of the original family Enterobacteraceae are placed in the new order. Enterobacterales is divided into 9 families, listed here, and it includes the following genera. So general characteristics are listed here: gram-negative, facultatively anaerobic, non-spore-forming bacilli and coccobacilli. They don't produce cytochrome oxidase except for Plesiomonas. They ferment glucose. They reduce nitrate to nitrite, with few exceptions. They're motile at 37 except for Klebsiella, Shigella, and Yersinia. Except for Klebsiella, Proteus, and some Enterobacter isolates, none has remarkable colony morphology on laboratory media. They all appear large, moist, gray on sheep blood agar, chocolate agar, and most non-selective media. Most are non-hemolytic on SBA except for E. coli. So microscopic and colony morphology, again, gram-negative, they don't form spores. Facultatively anaerobe bacilli, coccobacilli, or straight rods. Colony morphology on non-selective media, they're of little value in initial identification. General macroscopic morphology, large moist gray colonies on non-selective media and are indistinguishable. The exception would be certain species of Klebsiella, sometimes Enterobacter. Selective and differential media are available for presumptive identification of clinical pathogens. This is a flowchart of distinguishing features of the various species. This is going to be really helpful when you start to prepare and study for not only the upcoming test, but also the qualifying exam. Virulence and antigenic factors, they're able to adhere to, colonize, produce toxins, and evade host defenses. Many pathogenic species of this order possesses antigens that can be used in the identification of different serological groups. 2 major types are opportunistic pathogens or primary pathogens, and there's 3 primary pathogens: Salmonella species, Shigella species, and Yersinia species. This is a list of the common species and the diseases that they cause. So this is a good quick reference for the different diseases that are caused. Antimicrobial resistance is a significant issue. So one of them as urgent is carbapenem-resistant Enterobacteraceae, CRE. This is serious and concerning, and if it's identified, it is reportable. It's resistant to imipenem, meropenem, doropenem, and ertapenem. Documentation that the isolate possesses a carbapenemase. Real-time PCR assay detects 5 genes for carbapenemases. Screening of hospitalized patients is recommended to help contain the spread of carbapenemase-producing bacteria. 2016 discovery of plasmid-transmissible colistin marker MCR1 in a CRE strain. Antimicrobial resistance continues to grow more serious. Example of strains that produce plasmid-mediated extended spectrum beta-lactamases. ESBL would be E. coli, K. pneumoniae, and K. acetoca. So first we're going to talk about some of the opportunistic members. E. coli is considered opportunistic. Most strains are lactose positive. Beta hemolysis on blood agar plate. Green metallic sheen on eosin methylene blue agar. Motile and possess adhesive fimbriae and sexpili. Commonly isolated from the colon biota, and it's used as a primary marker of fecal contamination in water quality testing. Biochemical reactions: fermentation of glucose, lactose, trehalose, and xylose, indole production from tryptophan, glucose fermentation via mixed acid pathway, so MR positive, VP negative, does not produce H2S, DNase, urease, or phenylalanine deaminase. IMVIC pattern: IMVIC would be indole, methyl red, Voges-Proskauer, or citrate. Indole positive, methyl red positive, Voges-Proskauer negative, and citrate utilization is negative. This IMVIC pattern will allow you to determine different test results, or knowing these results will help you distinguish the various species, and an IMVIC pattern is going to be extremely important as we start to talk about the different species and be able to identify them based on our laboratory results. It cannot use citrate as a sole carbon source. The growth of E. coli on the various plates, you can see all of these are listed here. And you can see on McConkey agar that this plate it's growing pink because it is a lactose fermenter. You can see E. coli on these plates as well. Extraintestinal E. coli infections, one of the most common causes of septicemia and meningitis in neonates. Some strains have the capsular antigen K1, documented virulence factor, high fatality rates. Similar infections are uncommon in older children. In adults, bacteremia as a result of urogenital infections from a GI source. Other sources would be colonization of birth canal just before or during delivery, contamination of the amniotic fluid. Septicemia and meningitis are most common in neonates and very young children. They gain infection just before or during delivery and when there is contamination of amniotic fluid. Uropathogenic E. coli, this is the most common cause of UTIs in humans. It's primarily the E. coli that came from that person's own intestinal tract that happens to get into the urinary tract and cause infection. Cytolysins or hemolysins can kill immune cells and inhibit phagocytosis. Gastrointestinal pathogenic E. coli, there's 5 major categories that we're going to go through. Enterotoxigenic, ETEC, enteropathogenic, or EPEC, enteroinvasive, EIEC, Shigatoxin producing, STEC, exteroaggregative, EAEC. These 5 categories are sometimes collectively referred to as the enterovirulent or diagenetic E. coli. It's often difficult to distinguish diagenetic strains of E. coli to strains that are part of the normal microbiota by culture. So this shows some various features, such as virulence factors, the relevant disease, serotypes, laboratory tests used to distinguish them. So starting off with enterotoxigenic, or ET E. coli. Diarrhea in infants and adults in tropics and subtropics. This is known as traveler's diarrhea. It's transmitted by contaminated food and drink. Organisms can colonize the small intestine. Clinical manifestations include mild, self-limiting, watery diarrhea, abdominal cramps, occasional nausea, no vomiting or fever, and like other diarrheal illnesses, must be differentiated. Typical diagnosis includes characteristic symptoms and isolation of solely lactose fermenting organisms on differential media, immunologic assays, and multiplexed PCR. Enteropathogenic E. coli, associated with severe diarrhea in children younger than 1 year of age, nurseries, and daycares. It's rare in adults. Symptoms include a low-grade fever, malaise, vomiting, and diarrhea, persistent watery diarrhea, large amounts of mucus without apparent blood. Detection is based primarily on the suspicion of the physician. Entero-invasive affects adults and children, but it's rare in the United States. Dysentery with direct penetration, invasion, and destruction of intestinal mucosa. It's very similar to Shigella, but will require higher amounts of the inoculum. So in order to get this infection, you have to have like 1 million bacterial cells before you'll actually start to have symptoms, while Shigella has a much lower infectious dose. Clinical infection symptoms: fever, severe abdominal cramps, malaise, generally does not ferment lactose. Then there's Shigatoxin producing E. coli, or STEC. STEC refers to E. coli strains that produce Shigatoxin 1 and or Shigatoxin 2. Toxins are named because of their similarity to Shigatoxin produced by Shigella dysenteriae type 1. Shigatoxins preferentially affect endothelial cells of the GI tract and renal glomeruli. In 1982, the O157H7 strain of E. coli was first recognized during an outbreak of hemorrhagic diarrhea and colitis. These isolates are referred to as enterohemorrhagic E. coli, or EHEC, which is a subset of STEC. EHEC is clinically significant and reportable. If this is identified in a laboratory, we do report this. It's associated with hemorrhagic diarrhea, colitis, and hemolytic uremic syndrome. Hemolytic uremic syndrome is characterized by low platelet count, hemolytic anemia, and kidney failure. Classic illness caused by EHEC: watery diarrhea that progresses to bloody diarrhea with abdominal cramps and low-grade fever. No leukocytes are in stool samples. Symptoms range from mild to life-threatening hemolytic uremic syndrome. It can be fatal in young children and older adults. Mode of transmission: primarily processed meats, undercooked ground beef, unpasteurized dairy products, apple cider, bean sprouts, spinach, romaine lettuce. Laboratory diagnosis: stool culture on selective and highly differential medium with subsequent serotyping. Screening stool filtrates for the toxin and demonstrate a 4-fold increase in toxin neutralizing antibody titer. Culture media choices: so stool culture should incorporate McConkey containing sorbitol instead of lactose. SMAC with cefazime tellurite. Some strains are sensitive to tellurite. E. coli 01577 or -H7 strain does not ferment sorbitol in 24 hours, so that's why it's included on the medium as a differential characteristic. And this is a key point for this type of E. coli. Enteroaggregative E. coli: the cells stick together, cultured on cell monolayers, plastic surfaces, and the surface of the intestinal mucosa. Stacked brick pattern of cells between the cells by means of fimbriae can lead to biofilm formation. Symptoms mostly in children: watery diarrhea, vomiting, dehydration, occasional abdominal pain, persistent for at least 2 or more weeks. So switching gears from E. coli to Klebsiella. Klebsiella is found in the intestinal tract of humans as normal microbiota. It is associated with opportunistic and healthcare-associated infections, particularly pneumonia, wound infections, and UTIs. Common characteristics: most grow on citrate and in potassium cyanide broth. None produce H2S. Few hydrolyze urea slowly. All are methyl red test negative and Voges-Proskauer positive. With few exceptions, no indole is produced from tryptophan. So if we think about the IMVIC pattern, indole, methyl red, Voges-Proskauer, and citrate, it's negative, negative, positive, positive. So it's the reverse or the opposite of E. coli. Motility is variable, however, the predominant pathogenic species, Klebsiella pneumoniae, is not motile. Here is a table differentiating the species of Klebsiella. Klebsiella pneumoniae is the most common isolate respiratory tract infection of hospitalized patients. Also wound UTIs, liver abscesses, and bacteremia. Moist gray mucoid colonies, and the virulence factor is a capsule and that's responsible for making those colonies look mucoid. So this is a picture. They do take on that pink color and they look mucousy. Citrobacter consists of at least 19 species. They are inhabitants of the GI tract associated with infectious disease and healthcare associated infections. This most frequently causes UTIs. The species most often isolated are Citrobacter frundii and Casseri. Can be isolated from diarrheal stools. C. frundii has been associated with UTIs, pneumonias, septicemia, intra-abdominal abscesses, endocarditis in IV drug users. It's difficult to speciate Citrobacter. Most complexes hydrolyze urea slowly. They're positive for methyl red, ferment lactose, and grow on cinnamon citrate medium. And here's some differential tests to differentiate Citrobacter from Salmonella. Plesiomonas is formerly in the family Vibrionaceae. Oxidase positive, glucose fermenting, facultatively anaerobic, straight gram-negative bacilli, singly paired short-chain filamentous form, but there's no capsule formation. The genre Plesiomonas and Shigella share biochemical and antigenic features. Plesiomonas often cross-agglutinate with Shigella, hence the species name Shigelloids. P. shigelloides possesses a much lower virulence potential than Shigella, with low symptomatic carriage rates among humans. It can be serotyped by somatic O antigens and flagellar H antigens. They're found in soil and aquatic environments, widely distributed amongst warm and cold-blooded animals, and mode of transmission is undercooked seafood, particularly shellfish, in untreated water. 3 major clinical types of gastroenteritis: the more common or watery secretory diarrhea, a subacute or chronic illness that lasts from 14 days to 2 to 3 months, and a more invasive dysenteric form that resembles colitis. It's found in soil and aquatic environments, and it's widely distributed. 25 to 40% of all infected patients present with fever, vomiting, or both, and most common clinical symptom is abdominal pain. Most cases are self-limiting. Antimicrobial therapy is indicated in severe and prolonged cases. Morganelliaceae consists of 3 genera: Proteus, Morganella, and Providencia, widely disseminated in the environment and normal intestinal microbiota. It's recognized as opportunistic pathogens. All genera are non-lactose fermenting, and they deaminate phenylalanine. Proteus, there's at least 13 species. Mirabilis and vulgaris are widely recognized human pathogens. Both have been isolated from urine, wounds, and ear and bacteremic infections. The urease activity of Proteus mirabilis results in an increased urine pH and can lead to Sturvite kidney stones. This shows the swarming pattern of Proteus vulgaris on sheep blood agar. So you can see, even if you try to streak this for isolation, it's going to swarm and it's going to make this type of pattern. When we do certain types of antibiotic susceptibility testing, we usually create this thing called a purity plate. And a purity plate is just a streak of that organism to keep it pure so that we can compare to make sure that only one organism is growing on that plate. With Proteus, we have to make its own separate purity plate. So this is commonly encountered in the clinical lab, and we know that if it's a suspected Proteus species, we have to put it on its own plate because it will swarm and it'll swim all over the entire plate and take over the whole plate, and it will contaminate any other bacterial species you try to grow on that purity plate. So these are some differential tests to differentiate Mirabilis from vulgaris. Morganella is multi-drug resistant and increased virulence, healthcare-associated pathogen, UTIs, occasionally wound infections, and bacteremia. It's a non-swarmer and it's motile. Providencia, there's 4 notable species. Both have high resistance to antimicrobials. These are some differentiating characteristics of Proteus, Providencia, and Morganella. Edwardsiella. Edwardsiella tarda is the only recognized pathogen in the Edwardsiella group. It's urea negative, lysine decarboxylase positive, H2S positive, indole positive, no growth on Simmons citrate medium. E. tarda follows the same pattern of IMVIC as E. coli, so it's positive, positive, negative, negative, but this one's H2S positive, which easily differentiates it from E. coli. Clinical manifestations, bacteremia, GI infections, wound infections, and it's reported to have a significant mortality rate. Serratia. So Serratia marcescens produces a characteristic pink to red pigment, and that's what you can see growing in like your bathtub at home. If you have soap scum in your bathtub and you see like a pink-like color, that's typically Serratia marcescens that's growing. It's widespread in the environment, opportunistic pathogen, isolated from outbreaks in healthcare settings. So this is what colonies of Serratia look like. You can see that bright pink or red color. That bright pink or red color more predominantly grows at colder temperatures. So if we have it at room temperature, you're going to see a lot of pigment production. When you start to incubate them at higher temperatures like body temperature, 37 degrees Celsius, that pigment production decreases. These are opportunistic that are associated with outbreaks in healthcare settings, urinary and respiratory tract infections, nursery, cardiac surgery, burn unit outbreaks, contamination of antiseptic solution, and eye infections. So now we're going to talk about the primary pathogens: Salmonella, Shigella, and Yersinia. Salmonella inhabit the GI tracts of animals. Human infection occurs by contaminated or undercooked animal food products. Shigella, human carriers coupled with improper sanitary conditions or poor personal hygiene. And Yersinia is transmitted by a wide variety of animals. Salmonella is a gram-negative bacilli, facultatively anaerobic, clear, colorless, non-lactose fermenting on McConkey end colonies with black centers on HE or XLD. Negative for indole, phenylalanine, deaminase, urease, and VP. Most produce H2S. No growth in potassium cyanide. Classification: there's two species that are based on DNA homology and sequencing, Enterica and Bongori. Enterica is much more common and accounts for most, if not all, of human infections. And then there's 6 subspecies with Salmonella enterica. Virulence factors would be the capsule production, fimbriae, ability to traverse intestinal mucosa and survive inside of host cells. And then there's enterotoxins. Here are some differentiating characteristics between the different species. Antigenic structures. So primary somatic O and flagellar H antigens. O is heat stable, H is heat labile. You do need to be familiar with these antigens because a lot of our quick and rapid tests for Salmonella have to do with antibodies against these antigens. So there's the O antigen and there's the H antigen used in serological typing. Clinical infections: there's forms of salmonellosis such as acute gastroenteritis or food poisoning, typhoid fever, non-typhoid bacteremia, and carrier state. So starting with gastroenteritis or food poisoning. Sources of infection: primarily poultry, eggs, milk, egg products, handling pets. Pets such as reptiles, especially like turtles and tortoises, can carry salmonella. Ingestion of foodstuffs, calves and poultry petting at zoos, direct person-to-person contact, or cooking utensils. Symptoms appear 8 to 36 hours after ingestion of contaminated food. Nausea, vomiting, fever, chills, accompanied by watery or bloody tinged diarrhea and abdominal pain. Most infections are self-limiting. Antimicrobials of choice are listed below. Then there's the enteric fevers. Clinical features would be prolonged fever, bacteremia, and involvement of the reticuloendolar system, particularly the liver, spleen, intestines, and mesentery. dissemination to multiple organs. Enteric fever that's caused by Salmonella typhi is called typhoid fever. Transmission would be improper disposal of sewage, poor sanitation, lack of a modern potable water system. Epidemiology highlights tropical and subtropical areas where international travelers are more likely to acquire the infection. Symptoms develop 7 to 14 days after ingestion of organisms. The larger the inoculum, the shorter the incubation time. First week of disease, patients typically develop sustained fever, malaise, anorexia, lethargy, myalgia, and a dull headache. Organisms appear to be resistant to gastric acids, so they reach the small intestine, invade, and penetrate the intestinal mucosa. This leads to constipation. Organisms gain access to the lymphatic system and are sustained in the mesenteric lymph nodes. Organisms then reach the bloodstream and spread to the liver, spleen, bone marrow, and are immediately engulfed by mononuclear phagocytes. The organisms then multiply inside of your phagocytic cells. Then they're released into the bloodstream for a second time, and that's when the febrile episode becomes more evident. At this time, the organisms may be isolated from the blood. Weeks 2 and 3 of the disease without treatment, the patient experiences sustained fever with prolonged bacteremia. Organisms invade the gallbladder and Peyer's patches of the bowel, and they reach the intestinal tract by the biliary tract. Rose spots appear during week 2 of the fever. Infection of the intestinal tract typically transpires, resulting in large numbers of organisms. Invasion of the gallbladder and Peyer's patches releases bacteria into the bowel via the biliary duct. The gallbladder becomes the focus of long-term carriage of the organism. Salmonella bacteremia without extraintestinal foci of infection caused by non-typhoidal salmonella. 2 groups are at risk of developing this infection: young children and adults. And then there's the carrier state. So individuals who recover from infection may harbor the organism in the gallbladder, and that's where it can chronically be carried for a long time. Carrier state may be terminated by antimicrobial therapy if gallbladder infection is not evident. Cholecystectomy has been the only solution to chronic state of enteric carriers. So cholecystectomy is removing the gallbladder. Shigella, non-motile. Most are slow lactose fermenters, generally do not produce gas from glucose. No urease production, no H2S and TSI, no decarboxylation of lysine, cannot utilize acetate or mucate as a carbon source. S. sonii is positive for ONPG and ornithine decarboxylase. S. flexnerni is negative for these. This is the colony morphology and distinguishing features to differentiate Shigella species. Human and large primates are the only known reservoirs. Transmission is direct person-to-person contact, fecal-oral route with carriers as the source, transmitted by flies, fingers, and food or water contaminated by infected people. Personal hygiene is key. Groups at risk include children in daycare, individuals living in crowded and inadequate housing, individuals who practice anal oral sexual activity, known to cause outbreaks on cruise ships. So shigellosis, important cause of bloody diarrhea worldwide. Responsible species vary in epidemiology, mortality rate, and severity of the disease. The predominant isolate in US and Europe are Shigella sonii, followed by Shigella flexneri. Sonii is characterized by fever, watery or bloody diarrheal, and initial symptoms include high fever, chills, abdominal cramps, pain accompanied by tenesmus 24 to 48 hours after ingestion of organisms. So marked by penetration of the intestinal epithelial cells after attachment of the organism to the mucosal surfaces, local inflammation, shedding of the intestinal lining, and formation of ulcers after epithelial penetration. Clinical manifestations are caused by the penetration of gastric epithelial cells and production of toxins. Dysentery is caused by Shigella dysenteriae type 1. Patients experience severe symptoms. Bloody diarrhea progresses to dysentery within a few hours to a few days. Painful bowel movements, which contain predominantly mucus and blood, may become life-threatening as intestinal complications develop. So switching gears to Yersinia. Yersinia has 25 named species. Most are considered environmental isolates. 3 species are considered human pathogens. Yersinia pestis is the causative agent of the plague. Pseudotuberculosis and Enterolytica are known to cause sporadic cases of gastroenteritis and other types of infections in immunocompromised hosts. All 3 species are zoonotic agents, which means they can't be transmitted between animals to people. So Yersinia pestis, this is the causative agent of the plague, bubonic or glandular, septicemic, and pneumonic forms. Plague is transmitted through the bite of infected fleas. Pneumonic plague is transmitted through respiratory droplets. So what happens with the plague is, as mentioned, there's different forms. You can get the respiratory form, the pneumonic plague, directly from another infected person. So if a person is infected and you come in close contact with them, it's through droplet contact that you would get that in your respiratory system, and you would develop pneumonic plague, which is highly fatal. The other form is the bubonic form, and that's when you get bit by an infected flea. So there's fleas that will bite animals, and they will then jump off the animal and bite the human. And when they bite the human, they transmit some of that Yersinia pestis microorganism. Once it gets into your skin, it travels to a lymph node. And in that area, the lymph node will swell and form a hard, solid mass known as a bubo, which is why we call this the bubonic plague. Typically, the lymph nodes in the inguinal regions, so in the groin region, are impacted because most people that get bit by fleas, they get bit in the feet or in the legs. If you get bit in the arm, then your axillary or your armpit lymph nodes would swell. So wherever you get bit, that's where the lymph node activity is going to happen. Then those lymph nodes are going to swell and become solid. From there, they're going to replicate inside of the lymph nodes, disseminate further into the body, and eventually can lead to the lungs, where you would then transmit that through the pneumonic plague. The bubonic plague is better to start with if we have to start with the plague, because it gives you a warning sign. When you see those bubos, you go and get treatment. Pneumonic plague, you get flu-like symptoms, which rapidly progresses to someone's death. So it's much more quickly escalates to death. This is a Class A bioterrorism agent. And I just talked about these. Septicemic plague is when it becomes all over your body and it leads to bleeding, leading to a rash, extreme weakness, abdominal pain, and so forth. It is non-motile, gram-negative, short, plump bacillus. Bipolar staining at each end of the bacillus resembles a safety pin appearance. On qualifying exams, it will ask you which bacterium resembles a safety pin appearance. So, those key terms that you have to remember as species related, that would be Yersinia. Grows on routine media, optimal temperatures of 25 to 30 C. Yersinia enterolytica causes gastrointestinal upset. So it's transmitted by the fecal-oral route after ingestion of contaminated food, often pork, vacuum-packed deli meat, beef, lamb, chicken. Major concern, the organisms survive in cold temperatures, so food refrigeration becomes ineffective preventive measure, kind of like another organism we learned about earlier in the semester, Listeria monocytogenes. Most common forms of infection, like I mentioned, are gastroenteritis, but then there's also appendicitis-like syndrome, terminal ileitis, septicemia, arthritis, erythema nodosum. Acute enteritis often affects infants and young children between the ages of 1 and 5 years old. Stools often contain blood, usually mild and self-limiting. Appendicitis-like syndrome occurs primarily in older children and adults. The arthritis form, uncommon, extraintestinal form, usually after GI episode or an appendicitis-like syndrome. Yersinia enterolytica is a gram-negative coccobacillus, and it has that bipolar staining, preferential growth, 25 to 30 degrees, motile at 25 but not at 35. Cold enrichment can be used to increase recovery in fecal samples. And here is a table that allows you to differentiate the different species within the genus Yersinia. Laboratory diagnosis of Enterobacterales. Organisms must be isolated from a wide variety, or can be isolated from a wide variety of clinical samples. So it could be like urine cultures, wounds, it could be blood culture, it could be a fecal specimen. So the appropriate and intermediate transport media is needed for isolation. Isolation and identification, you have to consider the site of origin. Generally, enteric opportunistic organisms isolated from sites that are normally sterile are highly significant. There's also multiplexed PCR assays that can detect multiple pathogens in clinical specimens. So in other words, if we have a fecal sample, we can throw it in a multiplexed PCR, and we have primers that are designed to identify key sequences, or sorry, not identify, amplify key sequences for many GI pathogens. So we do one PCR in one tube on one sample, and it'll just amplify anything that it sees, and it can report out what species it is. Direct microscopic examination is important if it's like a sterile site. You wouldn't want to just look at stool, like a fecal specimen, under a microscope. That would give you no information. Direct smears prepared from samples containing endogenous microbiota, such as sputum, do not provide valuable information. Direct smear examination of stool, like I said, is not helpful. It may reveal the presence of inflammatory cells and blood, but in general, you're not going to be able to identify what species is causing the infection just by looking at a fecal sample under the microscope. For fecal samples, protocols and media should be available for Yersinia, Aeromonas, Vibriomonas, Plesiomonas. Laboratories should develop protocols for the maximum recovery of enteric pathogens. Media to consider: MacConkey, sorbitol MacConkey, HE, XLD, SIN2, and others as appropriate. We will get into more of this types of media and the distinguishing features of the enteric pathogens when we do our lab. Lysine iron agar and triple sugar iron slants screening. There's different results that you can see, and those are listed here. Helps us differentiate species based on their reactions. XE and XLD agars. Salmonella on chrome agar. So you need to determine if the isolate belongs to the order Enterobacter, gram-negative, oxidase-negative, except for Plesiomonas shigelloides. Always use young colonies from SBA. If they ferment glucose, reduce nitrate to nitrite, and molecular biology assays are now available. This is a schematic diagram that will be useful when you start drawing differentiating diagrams. And then serological grouping is important for Salmonella and Shigella. You can feel free to read through the points to remember.